Hello, everybody. Today we're talking about phylogenies. In other words, how do we reconstruct the evolutionary history of a species or group of species, and how do we determine the genetic relationships between them? So, this is an important question. It's becoming increasingly important when we look at conservation. One example that often comes to my mind, anyway, is this thing here. This picture is obviously some kind of dog, some kind of wild dog, wolf type thing. But its status pretty much hangs in the balance based on what we think it's related to. If you talk to some people, well, they'll tell you that it's a absolutely unique new species of wolf inhabiting the southeastern United States, whose range is now limited to a single refuge in North Carolina, the Alligator River National Wildlife Refuge. An American wolf, otherwise known as the red wolf for its red coloration. But if you talk to some other people, they'll tell you that this is nothing more than a remnant hybrid between coyotes and the gray wolves that used to live all around this continent. Now, it's important because there's only about 20 of them or so. And a lot of money is being put into preserving these species, or this animal. So, depending on the outcome of answering that question, it'll determine what happens to this thing. How much do we devote? How much money are we willing to give to protect it? Well, the studies are still not 100% conclusive, but it looks like there is something unique about this particular animal. So this isn't a phylogeny. It's showing the relationship between the gray wolf, the big timber wolf that we know of, that spans not only throughout northern North America, but also throughout Eurasia as well, and the coyote, the smaller prairie wolf, desert wolf, the one that inhabits the southeast, or the southwest, rather, the United States, which has steadily over time moved further and further eastward, hybridizing with anything that it can get its hands on, including, or paws on, I guess, including dogs, right? Coy dogs are a real thing. And then you have these in-betweens, the eastern wolf, up in places like Ontario in Canada, and the red wolf that we already talked about. Well, an endangered species is going to have a ton of resources to protect, it, whereas a hybrid, not so much. So the question still up for debate, but things are kind of going on as they always have been in terms of protecting this animal. But this is a great example of how the identity of a creature can really determine its conservation status and what happens to it in real life. So we've known things are related to each other for a long time, and we've seen similarities, and this was the basis for Carol von Linné, or Carolus and Linnaeus, as he called himself, to classify life on the basis of a hierarchy, which is essentially determined by the sort of likedness between species, right? So in case you don't know, there's a binomial nomenclature. It's like a unique identifying characteristic for every species. It consists of their genus and their species name. And as you move up, you get ever more inclusive groups, right? So species, genus, and then family, order, class, phylum, eventually the kingdom, and all of these things are eukaryotes, right? So here it is the example of a leopard. And you know that we fit into this as well. Homo sapiens is our species name. Our genus is Homo. Our family is the Hominidae. Our order is the primates. We're obviously mammals. We're vertebrates, which is a subphylum of the chordates. We're animals, and we're eukaryotes because we have eukaryotic cells, right? With all the characteristics of that. So it's been known that things are related for a long time. But beyond that, you kind of want to get a feel for what the history is like, and you have to account for all those things that have gone on. And that's where phylogenies come in, where you attempt to reconstruct the evolutionary history of a group of species or families or whatever you happen to be looking at. And you're doing this based on usually organisms you have available to you right now. In the perfect world, you might have fossil records and everything, but it's very, very difficult, right, to have a complete fossil record. Taxonomy is this act of ordering things and coming up with names, right? So Carl von Linné was the first taxonomist, right? And since then, a big part of taxonomy involves constructing phylogenies. And only when something is unique enough do we say that this thing is its own species or its own order or genus or something like that. So, what sort of things do we use to determine evolutionary relationships between living things? Here are three very different things, right? Here's some tulips, a mushroom, and a human being. Human beings, being animals, are more closely related to mushrooms. Plants are a little more divergent, right? One characteristic we can use is the presence of chloroplasts. Since plants have chloroplasts and fungi and animals do not, well, the evolution of chloroplasts puts plants into a separate category than the others, right? And that's what you see here, a branching between what eventually led to us and the mushrooms, and being apart from that led to the tulips. The bottom line is you can make these things, but it all boils down to characteristics. You have to find what those characteristics are that are going to help you discern whatever it is you happen to be doing. And therein lies the tricky part. So that's what we're going to be talking about today. But just to give an idea of how to read these things. So here's a phylogenetic tree. This is a fairly complete one for the carnivores, right? So as you go further back, you have the carnivores as the order, and then you see it branching into the three sort of big groups of the carnivores. You have your feline group, right, which ultimately will lead to your family Felidae. You have your Canidae, which are your dog-like creatures, right? So obviously coyotes and wolves, as shown here, but also things like bears and stuff like that. That's part of that too. And then you have the Mustelidae, right, which are the sort of the catch-all. Usually we think of weasels, raccoons, things like that, right? Well, otters, badgers, that sort of thing. So who's more closely related to this coyote? The coyote being Canis latrans. Is it the badger or the leopard? Well, you probably know it boils down to the number of connections. And each of those connections is a unique event, right? The cat group split off first, right, and went on their own evolutionary trajectory, whereas the badgers, along with the coyotes, kind of stayed together and then had a later branching period. So if you were to trace back, you would find that their node would be the first to be encountered, right, between the badger and the coyote, which indeed are going to be the more closely related, at least compared to the leopard. So let's try this out. So here are some questions. So where's the common ancestor of A and G? So here's taxon from A to G. There's a common ancestor, which, as we say, roots the entire tree. And then each of these little branching points is a node. So who would you say is the common ancestor of A and G? Well, you probably noticed taxon A is on the side, taxon G is all the way on the other side. Where they come together is the place where it's called common ancestor, right? That's where they meet. Now, that's the case for A and G. If it was A and C, their common ancestor would be right here, right at the node where they meet. So which of these taxa are more closely related to each other? Well, when you look at the tree, there's ones that diverged, that were the last to sort of split in two, and that would be taxon B and taxon C, right? So they're the ones that were more recently diverged, and thus you would expect them to be more closely related, and you would expect them to be the most similar looking based on whatever characteristics you used to make this tree in the first place. Now, sometimes trees are unresolved, and there's situations where it's not quite clear where the relationships, how they fit in. These are called polytomies. There's an example of it here. Taxon D, E, and F, you notice they all come to the same point. Well, nature doesn't quite work that way. Things always branch off, right? So you're always going to have two choices, and there might be another choice soon after, but you're always going to have a branching into two and splitting into two. Whereas this sort of situation is an area that would require a little more work. Perhaps they need to use some new methods to resolve that relationship. And often in phylogenies and taxonomists, this is the thing that often gets people, that gets them into arguments and things like that. right? Now notice, these are just a bunch of words and lines, and there's no indication of time here. So you cannot use something like this to understand when things evolved. You can only understand the relative um, relationships between them, right? So you have no concept of time here. Are we talking in the thousands of years? Are we talking in the millions of years? What are we talking about? You can't tell. We'll talk a little bit later about things we can do for this. But let's give an example of where something like this could be applied, right? If you have a good phylogenetic tree, it's actually a pretty powerful tool. So uh, there's only two countries right now, and I believe Norway is the other one, but they have a sort of self-imposed moratorium on the whaling that they do. But the Japanese whaling program still continues. They're very limited on the number of species they can target. Antarctic minke whales are the main species, and even this is probably going to be regulated completely at this point. But they have a scientific collection permit. But in the past, and maybe to a degree now, I don't quite know. But often they would exceed the conditions of this permit, and sometimes would not only do it for scientific purposes, but also a lot of this whale meat ended up on the market, right? Where it's a pretty high priced delicacy. And this sort of whaling for commercial purposes was often done under the guise of scientific collection. The issue is whale meat looks the same. So you can't tell what species it came from. And there was evidence that they're not only targeting Antarctic minke whales, but also possibly targeting other species. So how could a good phylogenetic tree and understanding phylogeny inform a conservation case like this? Well, simple thing you can do is you can go and test whale meat, figure out what species it comes from, and you can do this by looking at, in this case, genetic relationships, but by using a well-known phylogeny and using markers, genetic markers, you can essentially assign them within a completed phylogeny. So here, for example, we have minke populations from the southern hemisphere as well as the North Atlantic. We have a humpback whale, we have gray whales, blue whales, very protected species as our fin whales down here. And then you have your unknown samples, which in this case would be that whale meat. Well, if you run it through the phylogeny and you find a place to place these unknown samples, you can figure out whether they're minke whales or not. And when you do this, well, you have some of them showing up as being closely related to humpback. You have other ones showing up as being closely related to fin whales. And this suggests that they're not targeting just minke whales, but also going after these other species, right? So this is an example where you're building evidence saying that, hey, you're exceeding the conditions of your permit. You can't be selling this type of whale meat, right? Because once you cut up a whale, you don't know where that meat's come from. So how do you build one of these? Well, one thing you want to do is figure out what your characters are that are going to be the most important things for you. And once you do this, you can start to rank your groups based on characters and whether or not they have them or not. Now, this is often the key because depending on the characters you use, you can get very different results. On the one end, you want to have characters that are really variable, right? That have different easily diagnosable sort of versions, if you will. And if you carefully define your character and get it down to a binary of they have it and they don't, that's very helpful. But you also want a characteristic that is variable to the level that you're interested in exploring in terms of their evolutionary relationship. So what do I mean by this? Well, if you're interested in looking at, I don't know, comparing a family of birds, well, the presence or absence of wings wouldn't be the most helpful characteristic because just by the nature of the fact that they're birds, they all have wings, right? You may not want to use a characteristic like, the number of eyes they have or something like that, right? Something that's inherent in where they are. It's too broad a characteristic. So what might you use? Well, you might use the design of their tail feathers, for example, if you want to distinguish between very different types of birds. Maybe some birds that tend to be more ground birds have sort of flatter feathers, whereas other birds would have more rounded feathers, more aerodynamic or something like that, right? So those are the kind of traits you want to look for. Now, here we're going to give an example of developing a character table based on these characteristics for the vertebrates. Now, the vertebrates are a very, very big group. And, here in this case, we have a leopard, a turtle, a salamander, tuna, fish, a lamprey, like a parasitic eel type thing, as well as a lancet. And we've identified a lancet as being our outgroup. So what do you think the point is of an outgroup? Well, you probably guessed the outgroup is usually the most, I hate to use this word, but primitive um, organism, right? And it's the one that would generally lack most of the characters that would define all the remaining creatures. And what it does is it basically roots your tree to where you can see individual characteristics or characters sort of appear in your phylogenetic tree. And as they appear, you're making that relationship between those groups ever more exclusive to the point where you're going to narrow it down, and then you'll have a nice order to it. So when you look at the characteristics, what are the characters that they picked? Well, this is a character table right here. On the left side, you have vertebral column. These are all vertebrates, right? And with the exception of our outgroup, they all have a vertebral column. So the lancet gets a 0. The rest of them get a 1. Then the presence of jaws. Well, lamprey have this interesting cyclostome. Their cyclostome is meaning that they have this sort of like a dermal attachment. It's actually quite terrifying looking. But it's not a hinged jaw like you would think of. So it's not a mouth that can open and close the way we think of it, right? So they lack that. But the other vertebrates, with the exception of the lamprey, do have this characteristic. So lamprey gets a 0, lancet gets a 0, and the other ones get a 1, right? So there you're seeing, okay, the hinged jaws essentially evolve after lamprey, and you start seeing it in things like tuna and things like that. And you do the same thing for legs, the amniotic egg, a shelled egg. And mammals do have that. It's just internal. You have an amniotic sac. And then hair, right? Would define a characteristic of mammals. When you order all these like that, that character table can now translate into a phylogenetic tree, and you can see where all those individual characteristics show up on the phylogenetic tree. Right? So it's a very powerful tool. This is obviously a very straightforward example, but you can imagine that a big part of this depends on the characters that you choose. So the field of doing this is called cladistics. And in cladistics, your goal is to come up with a very good rationalized understanding of how a group of species are related to each other. And in order to do this, you need to produce a clade, meaning that you need to not only have to be able to explain everything that's in your study group, right? Let's say you're interested in blackbirds, for example. Okay, you're going to do blackbirds. You're going to have all these different species of blackbirds in your phylogenetic tree. Well, you want to see how they're all related to each other. So you want that tree to essentially be very much all of them coming to a single common ancestor. And when you do that, you have a common ancestor and all the descendants accounted for. That's a clade. So here are 3 groups, right? Group 1, Group 2, and Group 3. They vary. One of these is a real clade. Do you have any idea which one it is? Well, I'm hoping you picked Group 1, because Group 1 is an example of a monophyly or a monophyletic group. In a monophyletic group, you have a single ancestor, hence the mono, right? So you have a single ancestor, a single point of origin accounting for A, B, and C. We know how they're related to each other, and we get them down to a single point. So that's the goal of every taxonomist, is to come up with one of those. There are situations, however, where you don't have enough information. When you don't have enough information, well, it could be that you have a group that is either missing or an organism, a species that is either missing from your analysis or an organism that shouldn't be in your analysis and is counted anyway. So when you have almost everything, but you're missing something, you don't have all of the descendants from a common ancestor, we call that a paraphyletic group. Right, so that's group 2's example, right? So D, E, and F in that example, they're inside your group, your study analysis, but you're missing G. Now, G could be a living animal, it could be a fossil, right? It could be a fossil that just hasn't been discovered yet, but you suspect is there. Now, you have a situation like group 3, where you have groups of creatures or species that's included in your analysis that actually is not related to your, or not as closely related as you want to the rest of your organisms, right? So this is an example of a polyphyletic group. So just like a monophyletic group has a single origin, a polyphyletic group has multiple origins. So in this case, D, E, F, and G represent a nice monophyly, a nice clade. But by including C in your analysis, well, you now have this other thing that's not included, right? That shouldn't be in there, but is. So this is usually something that causes a lot of issues, right? And a lot of taxonomists argue about, oh, you should include C, or you shouldn't, because it's because of the characters that you use. That's why you chose the wrong characters in building your tree, and that's why C is shown that way. You should use this character instead. So this is where the sources of arguments come from. I know it's a little bit silly, but there are groups like this out there. So for example, if I say badgers, right? If I tell you the word badger, you're probably conjuring up what these images show you here. Small, short-legged, tough creature. It's kind of no nonsense, doesn't take no shit, right? That's the kind of creature you're thinking about. And on the surface, they look pretty similar, right? They're short, they're stout, they tend to be burrowing animals. They're obviously all carnivores. They have a lot of similar markings, right? But you have here an American badger, which is baring its teeth over there on the left. You have a European badger in the middle, which is sort of your classic sort of stereotypical badger. And then you've got the infamous toughest thing on this African savanna, the honey badger. And, it's surrounded with all these quills here after running with a porcupine. Just a tough creature. Now, we call these things badgers, but they're actually not related to each other as closely as you would think. Honey badgers are more closely related to skunks, as are other things like hog badgers, for example. Whereas, American and European badgers are only slightly related to each other. So by using the term badger, you're including a whole bunch of things that have different origins or are polyphyletic. So badgers are an example to me of a polyphyletic group. Right. So these are what phylogenies looks like, and these are the sources of kind of issues that taxonomists try to deal with. Now, one thing I'll mention is you can actually calibrate your tree based on time. And the way you do this is by having what's known as a molecular clock. I'm not going to get into detail about it, but a molecular clock basically gives you an idea of the amount of time it would take to build up mutations that would cause the differences between groups. And you can essentially alter your existing tree to show this, to where relatively short branches represent short spaces of time, whereas longer branches represent longer periods of time, more divergence. Right? And we'll get to mutations here in a bit. But the idea is that branch lengths are going to represent genetic changes as a result of these mutations. You could do this with a molecular clock. You can also do this with a fossil record if you have that as a good sort of overlay. If you don't have those two things, though, it's very difficult to make this kind of a phylogenetic tree. But here's a phylogenetic tree showing animals, humans and mice diverged from each other during the Cenozoic, the most recent era, whereas you go further back into the Paleozoic, that's when you see the divergence between chickens and mammals, birds and mammals. That's synapsid sauropsid split, goes far back there. Don't worry about that term right now. So The idea, though, is you're trying to build trees that have information attached to them as well. All right. So how are things related to each other? Well, there's two types of characters that you can use. And we identify these as plesiomorphies and apomorphies. Plesiomorphies are characteristics that are shared between groups because an ancestor of those two individuals or those two groups had this characteristic. So it's something that's inherited, right? It's an ancestral trait. That's what a plesiomorphy is. In contrast, an apomorphy is a characteristic that is, as we say, derived. And it's derived in the sense that this is a novel trait that evolved and is often relegated to identifying a particular group of organisms. So birds, for example, the defining apomorphy of a bird is the feather. All birds have feathers. Nothing that, there's nothing that is not a bird that has feathers, right? So, the apomorphy for the birds is the evolution of the feathers. That's their shared derived character that defines all birds, right? Whereas birds also have limbs, right? They have four limbs, right? They have their wings, but they also have their two legs. That is a characteristic that is not unique to birds, right? Reptiles have that, amphibians have that. So that's an example of a plesiomorphic trait. Right? So apomorphic trait is the shared derived unique thing. Plesiomorphic trait is that ancestral thing. So if you're building a phylogeny and you want to define a clade, what's the most useful thing? Is it going to be an apomorphy or is it going to be a plesiomorphy? Well, hopefully you guessed apomorphy. An apomorphy is going to help you define your clade because you can define your clade based on when that trait shows up. So all of the birds represent a clade when you look at feathers, right? As soon as the feather shows up, anything past that point is a bird, right? Whereas, it could be hair for mammals, right? It could be something like that, right? Speaking of mammals, here's a human being sitting pondering life. Here's a gelada baboon from Ethiopia, from the highlands of Ethiopia. Very cool. One of the coolest things I've ever seen in the wild. That's also chilling and contemplating life. What might be an example of a plesiomorphic trait between these two, and what might be an example of an apomorphic trait between these two? Well, plesiomorphic trait, there's no shortage, right? Limbs, facial features, two eyes, one mouth, nostrils, nails, name it, right? Very similar anatomy between these two because they're both primates. So any number of plesiomorphic traits. An apomorphic trait, well, one defining thing about baboons that apes like humans don't have is they have that little cushion on their butt, right? It's called an ischial So that's an example of an apomorphic trait that's unique to baboons in particular, but those old world monkeys in particular. Baboons also have a tail. That could be considered an apomorphic trait, although humans kind of lost their tail. They sort of have it, but they don't. But either way, for this example, you're understanding, right? So an apomorphic trait is something that would be unique to baboons or unique to monkeys. So let's move on. So sometimes it's a little more complicated. Here are two eyes. One of them is the eye of an octopus. One of them is the eye of a human being. This is one of the tricky things about phylogenies. Any idea which is which? Well, so both octopus and humans have essentially the same kind of eye, what's known as a camera eye. It works by having light go through a lens, which is then focused on the back onto a layer known as the retina. The difference between humans and an octopus boils down to the distribution of receptors. In humans, the receptors are located behind the nervous tissue, and the nervous tissue all comes together at a point and produces a blind spot. Whereas in octopus, they have the actually more rational version where light hits the retina directly, the photoreceptors, your rods and your cones, and then it gets sent through the nervous system that's located behind them. And the result is you don't have a blind spot. In case you're curious what the blind spot is, it's number 4. So the less efficient, kind of bizarre, backwards human eye is the one shown on the left, whereas the frankly more efficient, more rational looking octopus eye is the one on the right. So very, very similar, very small difference between these two. But the fact that they both evolved a very similar eye, well, there's a lot of things we could say about that, right? Maybe Davy Jones was correct. Any of you Pirates of the Caribbean fans out there, right? So his eyes might even be better than ours. So crazy thought, right? But this is an example of one of the sort of pitfalls of constructing a phylogeny. And that is identifying what is an analogy versus what is a homology. So an analogous structure is something that is very, very similar between two groups, but they're similar because of the similar sort of pressures being placed on those creatures that are then going to evolve the same situation, right? Sort of same response, not situation, response, right? Whereas a homology is an example of a trait that's just modified through time to take different roles. So one analogy is the environment imposing restrictions on what can evolve, and the result is they evolve very similar things, whereas a homology is environment acting on the same thing in different ways to produce different outcomes suitable for each particular situation. So here's an example of two creatures that live very similar lives, right? You have, everybody knows what a mole is. A mole obviously burrows in the ground. There's a lot of similarities between the North American mole, which is kind of your true mole, versus the marsupial mole, which is a marsupial, which should tell you that it's more distantly related. It is a mammal, but they're on sort of opposite ends of the spectrum in terms of evolution, right? So they're distantly related creatures, and yet they look very similar. Right? They're stout, they have small hind limbs, very large forelimbs for digging. Their heads are pretty large. They've reduced their eyes. They have a very large nose that they use for feeling their way through the ground. So very similar conditions, very similar lifestyles are producing the same outcome. So this is an example of, you know, all of these traits that I just mentioned being analogous between these two, right? They're similar because of the similar environment acting on them. But it's not because those traits were inherent that then got modified because of some common ancestor between them, right? There's a lot of other mammals in between a marsupial mole and a North American mole that's a placental mammal. So this produces, these analogous structures are the product of something that you probably know, convergent evolution. Convergent evolution is what creates these analogies, right? And this is tricky because something like this can be pretty well discerned, but sometimes you might have an analogy that arose because of convergent evolution and you don't quite know it. And the result is you don't know whether to use that characteristic to construct a phylogeny or not. And if you make the wrong decision, you could end up with a phylogenetic tree that is invalid. And this happens all the time, right? So it's very important to know what's an analogy versus what's a homology. Now, in terms of homology, you know homologous structures. They're actually very useful for coming up with phylogenetic trees as well. So if you can identify homology and discern the homology from an analogy, that'll really help you in constructing a tree. So important thing to remember when it comes to evolution, things are only going to change so much. There's a lot of inertia, phylogenetic inertia it's called, that's going to put a limit on what you can do. And often what you're going to see is remnants of past things that even if they're not being used now are still around or have been modified in some particular way. So developmentally, for example, human embryos and chicken embryos, they look very similar for a good chunk of their development. They have a lot of characteristics that are remnants of the fact that they used to be fish, right? Pharyngeal arches, pharyngeal gill slits, these are basically cartilaginous structures that support the gills, right? You know that humans don't have gills. You know that chickens don't have gills. Fish have gills, but they evolved from fish, and those structures are still there. And they eventually get reabsorbed, and they help to develop the jaw and stuff like that. Same thing with a tail. A post-anal tail, that's a defining characteristic, is along with the pharyngeal arches of the chordates. And for a short period of time during development, there is a post-anal tail in the human embryo, which then gets reabsorbed, right? Same thing with the chicken. These are remnants of the fact that they evolved from creatures that had longer tails. So these are examples of homologous structures in development. And then the classic example is the mammal forelimb, right? So you look at the human bones, the humerus, the radius, the ulna, the carpals, metacarpals, phalanges, all the bones of the arm. You see those bones slightly modified throughout the mammals, right? So cats have modified, especially their phalanges have been modified for running, right? The whale has modified the phalanges for producing a flipper. The bat has modified them for a bat wing, right? All mammals, all making do with the same bones. You know, in some cases they get promoted and become bigger and larger structures. In some cases they become not demoted, but become smaller or maybe fused, like you see in the whale with the radius and the ulna. They become very small structures, very stout, providing support for the flipper, right? So homologies can be very useful. And they can be very useful, especially when you want to look at fossils, right? So here's another classic example where you can look at the relationship between living creatures as well as their evolutionary past through fossils, and you can see the development of these characteristics that can help to provide some idea of how things are related to each other, right? So here's it's not a tree. I actually cut the tree out to put that little statement up there, but you have ungulates, you have hippopotamus, and something like a hippopotamus was probably the first in the line that eventually led to the cetaceans or the whales. So Pachycetus is kind of a rat-like thing, but it took to the water, and you can see that its pelvis and hind limb bones are pretty comparable to any other kind of running mammal, right? But over time, you see this reduction. The pelvis becomes larger. You see a reduction of the foot, a reduction of the tibia and the femur, eventually to where you get to living cetaceans that basically have a pelvic remnant, right, and have lost those other bones, right? So these are examples of homologous structures being used to reconstruct sort of the evolutionary history. So it's great if you have fossils, but what happens when you don't have fossils? Well, nowadays, a good chunk of taxonomy involves genetics and looking at molecular homologies. So DNA bases, you know your DNA bases, you know that A binds to T and G binds to C. You know that they line up the way they do based on that relationship. But you also know that this can change, right? And there's one thing in particular that's constantly happening to change the DNA sequence. And you know what that is, right? Of course you know what that is. There's examples of it on this slide, right? We're talking about mutation, people, mutations. And mutations can come in any number of ways, right? You can delete things. You can take out nucleotide bases. You could add in nucleotide bases or insert them. And every time you do this, if you look at a segment of DNA, well, you might be shortening it. You might be making it longer, right? You're changing the size of that. But if you account for these deletions and insertions, you can usually still line up DNA sequences between two individuals, between two organisms, right? And if you line them up, you'll end up having gaps, right? And I'm sure you can understand, the more closely related something is, the fewer mutations have happened in between them. So those nucleotide sequences are going to be lined up a little better. There'll be fewer gaps, right? So here again, you have a species A, or species 1 and species 2, right? Species one gets a deletion mutation, species two gets an insertion mutation. So now you end up with segments of DNA that are different lengths, right? And this could be especially important if it's a gene, right? A lot of what they do to assign taxonomy nowadays, they use non-coding areas because you don't necessarily want selection. You kind of want something in Hardy-Weinberg equilibrium, ideally. Um, but you could get, if you are looking at genes, you could end up with genes of different lengths. And what would these be? What is being shown here? A short gene versus a longer gene? Well, these are different alleles, right? These are different alleles. And even though they're different lengths, there's still going to be a lot of overlap. And you can use really powerful computers that run for a very long time, and they'll run multiple versions, 10,000 iterations of this stuff. And what they'll do is they'll actually align your sequences for you. And when they do this, then you can identify where those gaps are. And once you've done that, well, now you have a quantitative way of assessing relatedness. So distantly related organisms are going to have a lot of gaps, right? And their sequences are going to be very different. And where they do match up, well, it's thought that, you have about a 25% chance of matching up at any particular spot, simply from the fact that you have only 4 nucleotides to choose from, right? You're only going to have nucleotides. So you're only going to have A, T, G, or C. So there's already about a 25% chance that at any one spot, you're going to get the correct match, right? Just like in a multiple choice question, right? If you just go completely random and you have 4 choices, you got a 25% chance of being right on that multiple choice. So the more distantly related you are, the more gaps there's going to be, the less lining up of the sequence there's going to be. More closely related, the fewer gaps there's going to be, the more similar the lining up's going to be. So there are some rules that we use to assign this. One is this idea based on Occam's razor. Occam was a theologian and philosopher from medieval Europe. I want to say Germany or Poland, Prussia at the time. I don't remember exactly. Escapes me. But he had a very straightforward idea. The fewer assumptions that you need to make is going to likely be the correct answer, right? So when you have 2 potential explanations for something, the one that's going to be more likely is the one where you have to assume less things, right? So that's what we know as Occam's razor. And Occam's razor is a defining principle for maximum parsimony. And maximum parsimony is saying, okay, when you're lining things up and you're trying to build a phylogenetic tree, the one that's got the fewest steps in terms of evolution, the one that had to evolve the fewest apomorphies, is most likely the correct one. So what you're saying is, if you've got divergence between 2 groups, well, the relationship that requires the fewest evolutionary sort of things to happen along the way are going to be the most likely to be correct, right? It's basically fewest assumptions. So this maximum parsimony is especially when it comes to DNA, it's sort of reconfigured into a principle of maximum likelihood. And it's saying that, okay, there's certain assumptions you can make about DNA mutations, right? For example, if you have an insertion of DNA, well, you have an equal chance of getting an A in there, a T, a G, or a C, right? If you're getting a deletion mutation, there's an equal chance that all of, any one of those can get the deletion equally, right? So there's not like one thing leading to one being more favored over another, right? So when you have that idea in terms of like your mutations, for example, you find that, okay, the DNA sequences should line up with should be reflective of the tree that will work will be the one that has the fewest branches coming off it. That is most likely in terms of evolutionary changes, right? In terms of changing those DNA sequences. So the DNA sequences should be the minimal number of changes in those DNA sequences should produce the most divergence, right? So you don't want to start assuming things like, okay, you've inserted something, but then you deleted it later, but then you reinserted it. That's kind of terrible, right? That's you're making extra assumptions. So those are the sort of defining things. Now, these are tricky. They're not something that taxonomists necessarily worry about. And frankly, there's computers that do this stuff now. So what they'll do is they'll run, when you're doing an analysis like this, you'll produce like 10,000 different versions of a phylogenetic tree for your data. And the one that is the strongest one is the one that's going to kind of rise to the top in terms of its ranking based on these principles of maximum likelihood. So it's an interesting thing. It's quite interesting to see these computers run. They just run all night. They just kind of set it up, run it, leave, come back the next day, and it's like, oh look, there's some data. Pretty cool. So here's another sort of maximum likelihood assumption, right? So you have three organisms. This is showing in this top table here. It's showing humans, mushrooms, and tulips. Again, that example. And it's showing the percent divergence in DNA between these groups, right? So between the human and the human, there's no difference, right? Obviously. But between the human and a mushroom, there's a 30% divergence in their DNA. Whereas the human and the tulip has a 40% divergence in their DNA. So if you follow this table, well, 30% divergence, you can produce, here's an example with two trees. Which of these two trees is going to be most likely? Tree 1 and tree 2. Okay, well, hopefully you probably remember the picture I showed you earlier, the same tree, right? So tree 1 is the one that's most likely. But let me explain why that is. So when you look at tree 1, the difference between humans and mushrooms, the percent genetic divergence, instead of a 30%, has 15% on both sides. Obviously, 15 plus 15 is 30%, right? But what it's saying is, okay, when that split happened, the lineage that led to humans underwent the same amount of genetic divergence, the same number of mutations, right? It accounted for the same percentage of that difference as when that common ancestor split towards the mushroom. So both of them equally diverged. Both of them underwent the same number of mutations to produce a final outcome where they're 30% difference. 15% difference from the humans mutations, 15% difference from the mushroom lineage mutations, right? So that's an example of maximum likelihood. That's an example of the assumption, right? Same thing with between the whole group, right? It's a 40% difference between tulips and the others, right? The other two groups. Well, that 40%, well, 20% of that 40% comes from mutations that happened in the plant lineage that led to the tulips, right? And the remainder happened from the divergence that way, right? Towards humans and the mushrooms. But if you look at tree 2, it doesn't really make much sense, right? Because now all of a sudden you're saying, okay, 15% genetic divergence was needed to produce a human from a common ancestor with tulips, but for some reason the tulip had 25%, had a greater number of mutations that had to happen to produce a tulip. That's not quite what it works, right? Why would mutations favor the one lineage over the other when the assumption is that DNA is just DNA? When you boil it down to it, we're all just DNA, right? So when you have DNA, these mutations are going to be happening at the same rate in both lineages. So that's a little tricky. I definitely recommend you read up on this if you're interested. But the thing to remember about these phylogenetic trees is they're quite a pain to construct. You have to pick your right character. But once you have a good phylogenetic tree, it's basically a hypothesis that you can test, right? And especially when you look at fossil records and things like that, they're often missing or incomplete, right? And we definitely don't know about the behavior of, say, something like dinosaurs, for example, because we don't obviously they're not there. We can't observe them. We can't watch, videos of them, right? But you can make some assumptions and you can test hypotheses. And that's what a phylogeny is. So here's a phylogeny looking at the reptile clade. And notice that the reptile clade includes not only your classic lizard, snakes, and crocodile reptiles, but also includes birds here, right? Birds are considered reptiles. They're basically that lineage of the reptiles, sort of reimagined. And you've probably heard that birds are related to dinosaurs, and they're basically, the dinosaurs that made it, if you will. There's two basic groups of dinosaurs. You have Saurischian dinosaurs, which are things like your T-Rexes, but also things like your um, sauropods, your big brontosaurus type things. And then you have your ornithischian dinosaurs, which are kind of like a catch-all group of all the other kinds like hadrosaur shown here, or stegosaurus, things like that. Triceratops, those kind of dinosaurs. So the question is, okay, which ones are birds more closely related to? Well, this is a phylogenetic tree based on whatever characteristics they use. We know that birds are very closely related to crocodiles, right? We know that because they're around, and when you take a look at them, you look at their genetics, they match up very well, but dinosaurs have to fit into this picture too. So the question is, did birds more closely evolve from the one group of dinosaurs or the others? Well, it was reasoned for a number of different things, a lot of characteristics that Saurischian dinosaurs were the more closely related group, and together they form the clade of the birds. Well, if that's the case, well, now we can say, all right, if birds and Saurischian dinosaurs are so closely related, is there any evidence of, beyond just fossils, of any sort of behaviors? Can we make some assumptions about their behavior? Well, one thing that was made was, okay, well, birds are known for exhibiting really good biparental care of their offspring, right? They had nests, they guard nests, they incubate nests, they incubate the eggs. So did the dinosaurs do this too? Well, based on this sort of hypothesis, they must be pretty similar, right? Based on this phylogenetic tree. And when they went out and actually looked for remains in Mongolia, for example, they found many remains of oviraptor, um, basically sitting on eggs and fossilizing with eggs. And you can actually see the eggs in this fossil cast up here, and you see the bones of the oviraptor there. So this suggests that the oviraptor was very much like a seabird, almost. Like they would form these fairly small, loose colonies, and they would basically incubate eggs. Much in the same way they protect the eggs, much in the same way as birds do now. So it's quite an interesting thing, right? But that's an example of like, okay, let's see if we can find some evidence of bird behavior based on what we know about the phylogeny, that they're kind of related, right? And you would expect this more in the Saurischian dinosaurs than you would in the ornithischian dinosaurs. So dinosaurs lived a long time ago, right? Went out 65 million years ago, but 200 million years ago plus is when they actually evolved as a group, right? And that's a long time ago. And depending on how far back you need to go to answer your phylogenetic question, it'll determine the source of your DNA. So when you want to assign relatedness using molecular markers like DNA or RNA, for example, is another way to do it. You need to carefully think about what your time scale is, right? So, for example, if you want to look at the relationship between very divergent taxa or things that are thought to have diverged a long time ago, like for example jellyfish, which are cnidarians, from annelid worms like this nudibranch right here, right? That's a divergence that happened a long time ago, right? I want to say 375, probably more than that, right? I don't remember off the top of my head. You need DNA that's going to slowly accumulate mutations that will allow you to capture that divergence. So ribosomal RNA, the RNA found in ribosomes, that's something that the DNA that codes for that has a very low mutation rate, meaning that it's not going, the mutations will be there, but they won't be so many that they look completely different, right? It's that low mutation rate. So that'll allow you to answer a question like that. But let's say you're interested in more recent, right? In 100,000 or so years, as opposed to 100 plus million years ago, right? So you're interested in something that's more 100,000, 75,000, right? Well, you could look at mitochondrial DNA, which is inherited maternally. Mitochondrial DNA evolves very rapidly, and as a result, you'll be able to pick up more recent events. So if you're interested in, say, the relationship between creatures in North America versus Eurasia, and you're interested in the time of, when they were connected via the Beringia, the Bering land connection. If you're interested in sort of what happened then and then when it split, well, that's an event that happened with the last ice age. That's an event that you would need a more recent marker, right? So mitochondrial DNA evolves very rapidly. You'd probably be able to pick up a difference, right? And that's the kind of stuff that's used for Pleistocene data, right? So hopefully that's a good intro to phylogenies. Phylogenies do matter. One last thing I want to mention is sometimes when you have a gene, they can be very similar and they can be homologous, right? And they can share an evolutionary history. And especially when you think of the relationship between species, it can be very, very important to distinguish the difference between homologous versus analogous, right? But at the gene level. So genes can sometimes be very similar. Now, one thing about genes is they often duplicate. They often become different, and it's a very rapid event, right? So you can have a situations where, genes have undergone changes. Now, the thing to remember about genes is they're also doing functions too, right? They're usually involved in some kind of function, coding for some kind of protein that's doing some sort of thing, right? So you can sort of trace between species what that gene is doing and how it's operating, and also how that function can be controlled by multiple genes. Okay, so what the hell am I talking about? So here's gene A, for example, in the top picture. Gene A has undergone a duplication event, which has produced two versions of this gene, right? One's probably, a longer one, one's a shorter one. So now you have a gene A and a gene B, right? In the meantime, you have another species out there with gene C. Now, gene A, gene B, and gene C may all be doing the same thing, right? Same function, right? Maybe controlling some enzyme that metabolizes caffeine, let's say. So over time, species 1 gets a copy of gene A as well as a copy of gene B. And species 2 gets a copy of gene A and gene B. But because that species is also undergoing evolution, those genes are going to start to look a little different, right? They're going to start to accumulate mutations. So what you end up with is genes that might be doing the same thing. They're all metabolizing caffeine, for example, but they're going to look different when you look at their DNA and you look at the sequences of those nucleotide bases. So in species 1, well, they have gene A and they have gene B, right? Gene A and gene B are essentially related to each other because of that gene duplication event, right? So they're examples of what we call paralogs or paralogous genes, right? They have a common ancestry that comes from that gene duplication event. But in the meantime, gene A and gene B are both found in one species, and then gene A and gene B in species 2 are doing the same thing, but they've also evolved to be in a different species, right? So they've undergone their own mutations. So they're examples of orthologs to each other. So the gene A in species 1 is slightly different from the gene A in species 2. They're doing the same function, but they just look slightly different because of those mutations. So they're an example of an ortholog, right? And same thing with gene B in species 1 and gene B in species 2. They're orthologs to each other. So the differences between them arose from the speciation itself. So one arises from gene duplication, one arises from speciation. Now, gene C. Well, gene C belongs to some other species, species 3, right? And this is a gene that has nothing to do with the ancestry of gene A or B. But let's just say that it's also evolved to metabolize caffeine, and it's done so on its own. It's evolved independently. So that's an example of an analogy. So gene A and B are examples of homologous, because they're doing similar things. They're doing the same thing because of a common ancestry. But gene C is doing the same thing, but it's just because of, it evolved itself, right? It has nothing to do with a common ancestry. So gene C is just like that marsupial mole, right? That looks like a regular mole, so to speak. Looks like a regular mole, and in doing so, it's because of the same pressures. In this case, it's because of the same pressures to have something that metabolizes caffeine, right? So it doesn't have a common ancestor, but it does the same kind of job, analogous. So orthologs, paralogs, analogs. It's a good thing to know, especially as you move on to other classes in biology. But sometimes, especially with hybridization, you can get these different versions being dominant in a new species. And this is the case in Australia when a rainbow fish, the Van Heurus rainbow fish, which are these fish found in temporary ponds in Australia, they dry out, and they have to be able to tolerate very high temperature because their water bodies often end up being really, really hot. So this endangered species actually has an ortholog that it obtained, so to speak, its lineage obtained from hybridization in the past with another type of rainbow fish, a Bosemoni version of the rainbow fish, right? And the result is this ortholog, this hybridization event that happened a long time ago, that they now have this, has been under selection because it actually allows them to tolerate higher and higher temperatures, right? Which, as the world gets hotter, is going to be an increasingly important thing. So just like we talked about groveler bears and pisleys, and time will tell if this is a good idea, evolutionary speaking, it could be a similar thing happening with these rainbow fish in Australia. So again, phylogenies. They help to answer questions, and they help to provide information that can be useful for conservation. So stay tuned till next time. Have a good day.